Welcome back students to Yup Master, and now today's class we are going to study about the double membrane cell double membrane bound cell organelles. So these double membrane bound cell organelles are not forming the endomembrane system. In the previous class where we have concluded about the cell organelles of endomembrane system, there I told you that mitochondria, plastids are performing functions which may be related to each of them, but they are not at all forming a system. Means they can do the functions independent of one another. So that is why the thing that we need to know here, the double membrane bound cell organelles are not forming any system, and we are going to study them separately. So our first heading is going to be mitochondria. In today's class we are going to learn about the mitochondria, what functions are performed by mitochondria, and why the mitochondria is called as powerhouse of the cell. After this topic completion, means in the today's class, after completion of the today's class, you are able to compare mitochondria and the chloroplast, the two double membrane bound cell organelles, that how they evolved the life forms. So here we start with mitochondria. So the mitochondria, when we are going to study about where such kind of cell organelle is found. Mitochondria is the cell organelle that is performing aerobic respiration. So whether the plant is plant cell or its animal cell, if they are performing aerobic respiration, means if the cells are exposed to oxygen, in that cases these cell organelles are present in them. Although the mitochondria is absent, or mitochondria are actually absent in mammalian RBCs. The mature mammalian RBCs don't have mitochondria, and if a cell is not having mitochondria, it means it is not able to perform aerobic respiration. The number of mitochondria are determined by the metabolic or physiological state of the cell. If the cell is metabolically more active, it is going to have more number of mitochondria in it. Let's compare liver cell and muscle cell. Who is going to have more number of mitochondria in them? Definitely liver cell are going to have more number of mitochondria as compared to the muscle cell, right? So don't get confused that muscle cells are doing a lot of work, they are going to have more mitochondria. No, it's not like that. Okay, most of the metabolism in our body is taking place in the liver, and therefore the liver cell must have more number of metabolic enzymes in their mitochondria. Nextly, we say that how the mitochondria are originating. Just like nucleus is formed from the nucleus, cell is formed from the cell, the same way mitochondria are originated from the mitochondria. Means they are self-duplicating cell organelles. In short, you can write about them that these are self-duplicating cell organelles. Okay, and they get self-duplicated by a process that is called as binary fission, right? So this is about the mitochondria origin. So if we are saying the cell is dividing, the number of mitochondria are increasing. Okay, next about the structure. So mitochondria being a double membraned cell organelle, but it is still not visible if we are not going to stain it. Although in animal cell, we used to say in animal cell mitochondria is largest. Mitochondria, largest cell organelle. Okay, mitochondria is the largest cell organelle. And the next thing, if I am going to have the next thing, that after this, if plant cell is there, the number of plastid, the size of the plastid is greater than mitochondria. But right now we are talking about mitochondria only. About the plastid, we are going to mention such point later on. Although the name mitos means thread. Now what does a thread means? So thread is something like which is longer than the width. Means we are saying they are more longer than they are wider. So for example, here we say they are usually sausage shaped, where their average diameter is 0.5 microns and their length is almost double to 4 times, or 1.0 to 4.1 micrometers. So that's why we are saying they are appearing as thread like structure, right? Mitos means thread, and chondrio means something which is metabolically active. Okay, chondrium means something which is metabolically active. Fine. Next, about the structure itself, the appearance is like a thread or filament, longer as compared to as they are wider, and the size and the shape of the mitochondria depends on, we are saying it depends on what? It depends on the physiological state of the cell, osmotic concentration of the cell, particularly the proton concentration or H plus ion concentration. If they are saying specifically stained, the question is how the mitochondria get stained, what stain is used. So mitochondria is usually stained by using Janus green B, a dye which is used for staining mitochondria, or you can use crystal violet. Okay, or that you can use as crystal violet. Nextly, the other things about the structure itself, let's see in the diagram. We are going to discuss this diagram now so that we can easily understand what NCERT diagram is going to tell us. This is obviously appearing as a sausage, and when we are saying this sausage shaped structure, we have different parts. The first is outer membrane. Now what this outer membrane can do? The outer membrane is more permeable. We used to say the outer membrane is more permeable, allowing most type of substances to cross through it. The permeability is because of the presence of porins. Remember this term, porins. Porins are what? Integral proteins, integral membrane proteins that are present throughout the lipid bilayer. So therefore helpful in passing of hydrophilic molecules through them. Next, the second thing is inner membrane, obviously, and the inner membrane is comparatively less permeable, and this less permeable is because of the substance that is called as cardiolipin. So if someone is saying that what creates the barrier in the membrane, answer is cardiolipin is creating that barrier. Because of cardiolipin, the inner membrane is less permeable and it is having certain infoldings. You can observe these finger-like projections are here. Okay, here I can make, these are finger-like projection, these or infoldings. Now these infoldings are more prominent when the mitochondria is performing its function. What function? ATP synthesis. When mitochondria is performing ATP synthesis, at that time these infoldings or cristae are more prominent. Okay, so they perform ATP synthesis, remember this. Okay, and then comes the chambers, because there are two membranes. These two membranes are enclosing two chambers. The outer chamber, which is also called as intermembrane space or perimitochondrial space, whereas the inner chamber is obviously named as matrix. Now this matrix consists of the enzymes of Krebs TCA cycle, one of the step of aerobic respiration. So it means the matrix is having the enzymes of Krebs TCA cycle. Number two, the matrix is having 70S ribosomes in it. Means the matrix is having the enzymes of protein synthesis as well. And nextly, we used to say that this is having a single molecule of double-stranded circular DNA. Oh, the cell organelle which is having DNA. In that basis, we can say the cell organelle with DNA. Clear? These are known to create what? Or these are called as extra nuclear genes. These are called as extra nuclear genes. And remember, these extra nuclear genes are responsible for maternal inheritance. Means mother is going to pass on these genes to either of the progeny, means whether male or female. Extra nuclear genes are associated with maternal inheritance. The character from mother to son and daughter, both. But only daughter can pass these character to next generation, not the son. So remember the cell organelle with DNA. If any cell organelle is having the DNA, definitely it is going to exhibit certain hereditary characters. Okay, these extra nuclear genes may interfere with the nuclear genes or the usual genetic material as well. But don't consider them as a part of genome. They are not the genome. Genome is obviously confined to the nucleus, but the expression of genome sometimes may get affected because of presence of extranuclear genes like those which are present in mitochondria or in the plastid, right? Next, the other things about this structure which we have made, this is the sausage shaped structure which is representing the mitochondria. Okay, mitochondrion is best, the singular word. Okay, remember about the enfoldings that we have mentioned here. So here is the thing that we are discussing about how these foldings are. So these are enfoldings. Now these enfoldings, this side is perimitochondrial space, this is the matrix side. These enfolding is going to have some stalked particles. These enfoldings have some stalked particles which we often used to say as subunits of Fernandez and Morin. Clear? Often as subunits of Fernandez and Morin. So oxysomes, elementary particles, ET particle or F naught, F1 particles. Nextly, if this is head, okay, this structure is head. About these oxysomes I am saying the detailed structure, then stalk, then base. Now all of them have different roles. The base is going to act as proton channel. Interesting. The protons will come into the matrix from PMS, from here to here. Okay, who is going to allow these protons, the base, and this head is going to be the enzymatic type. This is enzymatic and favoring what? ATP synthesis. In total, the whole apparatus is working as ATP synthase. Okay, and then we used to say that this structure, this base, this stalk, and this enzymatic head, this is F naught base, F1 head. The protons are going from this side, means the perimitochondrial space, from their higher concentration to their lower concentration, and in this way they are making it favorable to form the bonds like this ATP, ADP to ATP. This is how we can mention them, right? So be sure about these things, that how we are mentioning these are two protons that I am telling you, and ATP is getting formed. This is just a brief description what oxysome has performed. That's it, nothing else, right? So here we are discussing about the function or how the ATP synthesis happened in mitochondria. Moreover, this ATP synthesis in mitochondria is dependent on presence of oxygen. Okay, I repeat, this ATP synthesis in mitochondria is oxygen dependent. So it is also named as, making of phosphate bond, phosphorylation, oxygen dependent, oxidative phosphorylation. I will say this all happened in oxidative phosphorylation. By saying oxidative, I am saying O2 dependent. Okay, more things to be discussed when we are going to discuss about respiration in plants, and in that chapter we have a more detailed study of this structure. Okay, this is the same oxysome. So remember, oxysome are not the cell organelle, they are part of cell organelle. Don't get confused. Okay, oxysomes. Next, mitochondria is gram-negative bacteria. Why I am saying that it is gram-negative bacteria, or what gram-negative bacteria is? So there are certain characters which are resembling in mitochondria and gram-negative bacteria. Like first one, the two are going to have porins in their outer membrane, due to which it is more permeable. Number second, multiply by binary fission. Both of them increases in number by binary fission. Number third, they have circular double-stranded DNA, means the genome you can say. 70S ribosome, which are the site of protein synthesis, and for protein synthesis the energy is provided by ATPs, which are synthesized in the mitochondria. So if any cell is capable, or if I am saying if any entity is capable to create its own replica, right? Binary fission to create its replica or its copy, and these all things for required metabolic activities. Try to recall the definition of unicellular cell organelle that we have studied earlier. Sorry, unicellular organism that we have studied earlier. What is the definition of cell? I said cell is defined in terms of unicellular organism. Now the unicellular organism is able to perform all the metabolic activities in the medium and is able to generate its replica in the medium. So the same is exhibited by the mitochondria as well. So can I call mitochondria as the individual cell? Answer is yeah, true, you can say this. So behalf of these things, where the structure is capable to generate its replica, capable to perform its metabolic activities in the medium, I am saying it seems that mitochondria are autonomous. Mitochondria are independent to the nucleus in the cell. Let's see, is it true completely? No, there is an objection. I said autonomous. Fine. Okay, but there is an objection. What objection? The thing is, the enzymes which are required for these metabolic activities requires enzymes. So if enzymes are required for these metabolic activities, the question is from where these enzymes are coming, or where the genes for such enzymes are located. Their genes are located in the DNA of mitochondria, yeah, plus nucleus also. Oh my, this is the thing. My God, the thing is that the enzymes which they require to fulfill their all metabolic activities are formed either in the mitochondria itself, or they are dependent on the cell to get those enzymes synthesized. So this is how mitochondria is not completely independent. It is sharing its life, or it is sharing the resources with the host cell. So it is not completely autonomous. I will rather say semi-autonomous. Okay, we can say semi-autonomous cell organelle. So why I am saying it is semi-autonomous? Why it is semi? The semi, half, is because of this line, that the mitochondria are not completely able to perform independent existence. They require a shelter, they require an association in the cell, because they require the enzymes which are synthesized by the nuclear genes. Okay, the same way, can I call it as a independent bacterial cell? Answer is no. Again, so I can say mitochondria is, or mitochondria are, better is are, although books are writing it as is mitochondria is, but mitochondria is a plural word. Okay, fine. So mitochondria are getting associated with the cell, they are performing a mutual interaction with the cell, they are giving ATP to the cell and getting the enzymes from the cell. So it is like that, a mutual association, symbiosis type of thing inside the cell. I will say are bacterial. Bacteria like why? 70S ribosome, circular double stranded DNA, binary fission, these type of thing. Bacteria. Bacterial character. 1, 2, 3, 4. Okay. So bacterial. Endo means inside. Symbionts means as a symbiosis. Clear? Bacterial endosymbionts. These will be bacterial endosymbionts, as I am stating here. So bacterial endosymbiont is something which is very clear to us, and these are the things that we need to mention. Okay. So, bacterial endosymbionts means they are symbiotic because they cannot perform all the metabolic activities outside the cell. They require enzymes which are made by nuclear genes. Okay. So, hopefully I think it is very clear to you why they are not completely independent, why they are not completely autonomous, because of their dependency on the nuclear genes. Although you can consider mitochondria as a prokaryotic cell, but not independent, the symbiotic type. Mitochondria are bacterial endosymbionts. Characters are mentioned. Okay, mitochondria are not autonomous, they are semi-autonomous. They are not fully independent of the eukaryotic cell. So just I am asking you, if they are something like a bacterial cell, then what kind of bacterial cell they are? Autotrophic or heterotrophic? And the answer is very simple. They are heterotrophic bacteria. Which type of bacteria? Heterotrophic. Why I am saying this? Because they are synthesizing ATPs from organic compounds. Mitochondria break organic compounds to obtain ATPs, and such is the habit of heterotrophic cell, not the autotrophs. Remember, autotrophs or producers are those which make the ATP by oxidation of inorganic compounds, and these are oxidizing organic compound to obtain ATPs. So, it's very clear. Remember about this. Functions. So, mitochondria is considered as powerhouse of the cell, and the reason is very simple. As I told you, they form energy currency of the cell. Now, what is energy currency of the cell? Energy currency is ATP. Energy currency is ATP. So, when I am saying mitochondria are powerhouses of the cell, they are making the energy currency or ATP. Number secondly, they synthesize amino acids. Number thirdly, they are responsible for extra nuclear genes, or we say that they are responsible for something which is mentioned as maternal inheritance. There is a separate topic, principles of inheritance, cytoplasmic inheritance. There we used to study about maternal inheritance, not here. So remember about mitochondria, how they are performing the functions. Because of maternal inheritance, even you can study the evolutionary patterns also. Mitochondria done. The next cell organelle is obviously about the plastid. Remember, mitochondria has taken in oxygen. Mitochondria has given out CO2. Mitochondria will do the gaseous exchange of which kind? They take in O2. They give out CO2. Okay, they take in O2 and they give out CO2. This is the thing that they used to mention about. Next, plastids. Now, plastid are another kind of double membrane bound cell organelle, the second type. These plastids are found in all plant cells, means eukaryotic one. We can see them from the members of algal protist to the kingdom Plantae, not in the fungi, clear, plant cells, plant kingdom and in kingdom Protista. Okay, these are easily observed under the microscope as they are large. So when I am talking about the plastids, remember, in plant cells we have something which is called as sap vacuole, which is going to occupy most of the volume. Then is plastid, and thereafter mitochondria. Remember, plastids are larger than mitochondria. Okay. Nextly, the other features, like they bear some specific pigment. If they have certain pigments in them, we can name them according to their pigments, and they are imparting pigmentation to those parts in which they are present. Okay. On the basis of the pigments, they are green colored plastid, chloroplast. Non-green but colored plastid, chromoplast, red, orange, blue, something like. Or the third type is non-green and colorless plastid, that is leucoplast. So these are three different type of plastid: chloroplast, chromoplast and the leucoplast. But wait, before I move to next, you must need to know that how these plastids originated. So plastids are going to show the interconvertible form. The three types of plastids are interconvertible. Clear? The three types of plastids are interconvertible. Fine. Going to the things. The first I have taken is chloroplast. So the chloroplast is something which is having green plast, which are green plastid having chlorophyll A and B with carotenoids in them. Chlorophylls provide the green color. Obviously, carotenoids provide something like red, orange. Okay, these type of pigmentation. When this chloroplast is going to lose loss of chlorophylls. Okay, this is loss of chlorophylls, and here we say chromoplast. So when we are talking about chloroplast, these are non-green colored plastids. If it is non-green color plastid, they have carotenoids in them. Okay, they have carotenoids, but they don't have chlorophyll. So remember, the chromoplasts are something which are orange, red type of colored, means they are providing pigmentation to flowers, fruits, sometimes variegated leaves also. Here it is. Pigmentation of flowers, fruits, sometimes condensed root, yeah, beetroot, carrot, they are colored because of certain type of plastids in them. Okay. Nextly, other examples, leucoplast. Say interconvertible. I am not going back from the chromoplast. The reason is simple. I am just taking the example of senescence. The color is changing, okay, because of the aging process. So leucoplast is colorless, non-green plastids, and they are performing storage. Reserve material. Leucoplast may get converted into chloroplast. Now that's possible. So they are interconvertible. I have a different word for it. You can call it as polymorphism also. Okay. Can you call it as polymorphism? Answer is yes. You can take it as polymorphism as well. Functional states are getting different. Here. Next. So first is the type which is called as leucoplast. We are going to study about leucoplast and chloroplast. Okay, chloroplast in much detail. Leucoplast. Now the leucoplast, what it is going to store is obviously responsible for the name. Amyloplast is storage of starch like in the tubers of potato. Oleoplasts are going to store oils or the lipids, as we see in many fatty seeds like castor, sunflower, groundnut, etc. Other example include alluroplasts, which are obviously having proteins in them. One such example you have studied, and that is allurone in maize grain. Clear? It is about allurone in maize grain. That's simple. Okay. This is the thing that we used to mention about alluroplast. So these are different type of leucoplasts. Remember, these are non-green, comma, colorless also. Okay. So that type of thing we used to mention about them. Next, the chloroplast. The chloroplast is about the occurrence. When we used to mention about the occurrence of the chloroplast, the thing is in plants that are exposed to the sunlight and air. Remember, sunlight, air. From air, what they need? They need CO2. Okay, from air they need CO2. From sunlight, what they are requiring? Synthesis of chlorophyll is light dependent. So the cells in the plant, if exposed to the sunlight and they are exposed to the CO2 also, then there is a chance that they possess chloroplast. I said there is a chance, not necessarily. Not all the export cells are going to perform photosynthesis. Particularly we say the mesophyll of leaf is going to have most abundant number of chloroplast. Thereafter you can find the chloroplast in ripening fruits, means they are green in color earlier, like green tomatoes turned into red. So they were chloroplast earlier, okay, or green chilies. Site of photosynthesis, they are the site of photosynthesis, they are going to fix the atmospheric carbon dioxide into sugar. These are the places where we say that, or these are the organelles which are not present in the meristematic cells. About the mitochondria I told you, when they are dividing, means what? The cells are dividing, at that time the number of mitochondria were increasing. But I cannot tell truly about the plastids, means I can't say that meristematic cell is having chloroplast. No, they don't have chloroplast. The meristematic cell is going to have something which is named as maybe proplastids, means the plastid whose fate is not determined or whose fate is not fixed right now. Maybe proplastids are present. It is possible they may have proplastids in them. Next, the thing is, so these are about the occurrence of the chloroplast, right? Remember this, you can take out a chloroplast when you are going to keep the green cell in a 10 molar solution of sucrose. You can extract out the chloroplast, you can study the chloroplast, and here is the thing which is chloroplast, how chloroplast looks like. So the chloroplast, just like the NCRT diagram, nothing different. See, here is the thing, outer membrane, inner membrane, these are same as we have studied in the cases of mitochondria. Outer membrane, inner membrane. There is a space between outer and inner membrane, and that space we can say as outer chamber. Wait, before that, this is granum, this is thylakoid. Now what is granum actually? Granum is nothing, granum is actually aggregation of thylakoid. And remember one thing, as I told you earlier when we were studying algae, at that time I told you that thylakoids are actually the places where the pigments, photosynthetic pigments are located. But these thylakoids were not associated or not forming the granum or grana in algal cells. Here they are forming the grana because we are studying higher plants right now. So the thing about the thylakoid, it is a place of photosynthetic pigments. It is the place of light reactions of photosynthesis. The light reaction of photosynthesis are something related to liberation of oxygen and synthesis of ATP. Now here this ATP synthesis in the chloroplast is light dependent. So being light dependent, so ATP synthesis is simple, phosphorylation. ATP synthesis is phosphorylation, and because light dependent, I will call it as photophosphorylation. Okay, this will be photophosphorylation then. So when we have these names, we are very clear about it. Photophosphorylation is the thing, okay, right? ATP synthesis will be seen in both type of cell organelles, whether mitochondria or chloroplast, but in mitochondria we studied the phenomena as oxidative phosphorylation. Here I am saying photophosphorylation. The two things are different, okay. Next, the thing is about the inner chamber, that is called as stroma. This pink line is showing what? This is the outer chamber actually, right? This is the outer chamber. Or intermembrane space. I can call it as periplastidal space as well. Periplastidal space. PPS, mitochondria PMS. The inner chamber in the chloroplast is called as stroma. There it was matrix. I can write for the stroma here as matrix of chloroplast. See the similarity in the matrix or in the inner chamber. Similarities. DNA molecule, 70s ribosomes. Now, enzymes of dark reaction, enzymes which are light and light independent. Clear? Light dependent, light independent, both type of enzymes are present. Similarity with the matrix of mitochondria is this: DNA molecule, 70s ribosomes. Okay, you need to remember about the similarities as well, the differences between chloroplast and mitochondria. So what we have learned here is about the characters that we need to study about the chloroplast. But wait, before we go further about the chloroplast, we need to know about functions. Okay, functions. What chloroplast can perform? The chloroplast is the site of photosynthesis, means carbon assimilation. Okay. Number next, the synthesis of organic acids. Number third, having extra nuclear genes, means again maternal inheritance or cytoplasmic inheritance. These all are the functions of chloroplast in general. All these functions we have mentioned. Okay. So the next thing about, are these chloroplasts can be compared with the gram-negative bacteria also? Yeah, they can also be compared. Before that, let's establish the polymorphism. As I told you that the chloroplast, leucoplast, chromoplast are interconvertible forms. So they are exhibiting the polymorphism. Examples, just tomatoes, they were having chloroplast earlier which get turned into chromoplast when the fruit get ripened. The same is about the roots of carrot. They were having leucoplast earlier which are getting converted into chromoplast. So the color is getting changed. This is an example, okay. These are the example by which we can say that yeah, these plastids are also exhibiting polymorphism. So before that, before this thing or before these plastids, we have studied polymorphism for two type of examples. Now, earlier lysosomes, and now it is about plastids. One is lysosome, other one is plastids. That's simple, easier one. So this is how we can explain, right? Coming to the next one, the chloroplast as gram-negative bacteria. Can I state these things? Yeah, obviously. We have certain statements which are going to be line to line same with the mitochondria. Like outer membrane is having porins. Number second character, they increase in number by binary fission. Number third, they have circular double-stranded DNA. Next, they are having 70s ribosomes and again ATP synthesis. Same thing, right? On the basis of these features, I can state it very clearly that these seems to be something as independent cell. They are able to generate their replica, they are able to generate their enzymes as well, right? The genes for enzymes are located in this DNA. The site of protein synthesis means the enzyme synthesis, ribosome. The energy required for the protein synthesis is from ATP. Everything they have, okay? But again there is a problem. For performing metabolic activities, they have all the apparatus, protein synthesizing machinery. But again the problem, the genes for these enzymes are present in, clear? The genes for these enzymes are present in what? The first, obviously plastid, no doubt. Number two, nucleus. Oh, I was about to state the chloroplast as independent as a cell, but still I am not able to mention this thing. Why? Because again the genes for the enzyme synthesis are located in the nucleus as well, or the genes for the enzymes they require to perform their metabolic activities are coming from nucleus as well. So they are again performing something equivalent to mitochondria, means a kind of symbiotic behavior, although they are different because these ATPs which they are using in protein synthesis and the other activities like photosynthesis are made in presence of light and by oxidizing inorganic molecules. This is a character of autotrophic bacteria, right? So chloroplasts are autotrophic bacteria type of things which are getting symbiotically associated with eukaryotic cells. Chloroplast is obviously like bacterial endosymbionts. So again, instead of writing them as autonomous, I will say they are not autonomous, these are they remain semi-autonomous. They are also not becoming completely autonomous. The reason is simple. Why? Because they are dependent on nucleus for getting the enzymes synthesized. Okay. So chloroplast is bacterial endosymbiont, and what type of bacteria it is? Obviously autotrophic. Mitochondria was heterotrophic endosymbiont. Chloroplasts are autotrophic bacterial endosymbionts. There are a lot more features that can be compared between mitochondria and chloroplast. I think or I hope that you are able to compare the chloroplast and mitochondria so that you can have a better kind of a good quality questions out of the comparison. This is what we need. Whenever you are reading something or some words which are common between two different structures, you need to do a comparison. Okay? When it is totally dissimilar, there is no need to do the comparison actually. You have studied 10 of the features, if 3 or 4 are similar in them, definitely it is going to be a question in the competitive exams. Hope you are following the classes well and attempting the daily practice paper as well. This is what we hope at Yup Master. Soon we are going to be Turito.com, and you are watching our classes live, as well as you can watch our classes in the recorded form as well. Just you need to download the Yup Master app, and there you can have the previous classes of the earlier chapter plus the content of the chapter. This is all for today. In the next class we are going to do a comparison, although I am giving you it as a homework, clear, the self-doing assignment, but I'll see whether you can do it yourself or not. I'm going to do the comparison from my side as well. Okay. So this is the thing that I need to explain in the next class, the comparison between the chloroplast and mitochondria as bacterial endosymbionts. Thereafter we are going to start with ribosomes and the cytoskeleton, the structures which don't have membrane around them. This is all for today. Thank you for watching the classes at Yup Master. Hope you are all doing well at your home and staying safe. Be regular in the classes, be punctual with the classes. Thank you. All the best.